We're here with Niels van der Lelie. He's the Senior Director for the Center for Agricultural and Environmental Biotechnology at RTI International. Niels, tell me a little bit about where you came from to RTI. Where were you before, and give me a little bit of what you were doing there.
Okay, so I come originally from the Netherlands. I have a PhD in molecular biology and microbiology, which was actually in the field of dairy production, more specific microorganisms that were involved in cheese fermentation. After that, I did a postdoc at Transgène in Strasbourg in France, which was a biotechnology company. Then I worked for 11 years in Belgium, first at the Belgium Nuclear Research Center and subsequently at the Flemish Institute for Technological Research, where my work focused on environmental remediation, both heavy metals and organic pollutants. And in 2001, both my wife and I were hired by Brookhaven National Laboratory in Upton, New York, and BNL is one of the national labs from the US Department of Energy. And there our work focused on heavy metals, and then very soon went to the growth of biofuel feedstocks on marginal land. And in 2011, January, I joined RTI as the Senior Director for the Center for Agricultural and Environmental Biotechnology.
And this is a new position, right?
Yeah, it's a new position. It's actually also a completely new center that RTI is creating. So far, RTI had an interest, especially out of the International Development Group, in agriculture as part of helping countries and their population to create a more sustainable livelihood. But we did not have here at RTI our own research in agriculture and environmental biotechnology, and this is something I'm setting up now.
Now, in the past, you've done some interesting things with microorganisms and plants, and you've had them do stuff that these plants and microorganisms usually don't do. Tell me a little bit about that.
So you're probably referring to our work on using endophytic bacteria to improve the properties of plants to deal with environmental contaminants. So endophytic bacteria are bacteria that are living inside the plants, and actually when you eat uncooked vegetables, fruit, etc., you're eating quite a high number of microorganisms at the same time, and a lot of people don't realize that. However, these bacteria are harmless and actually they're beneficial for the plant. You can compare them to the microorganisms that are living inside our gastrointestinal tract. And when we use these bacteria, it is like giving some of these yogurts with the probiotics that people are buying to the plants. So it's very beneficial. And what we did was we isolated bacteria that live, for instance, in trees like poplar, and then used genetic exchange between these bacteria and environmental bacteria that can degrade certain contaminants to create a new microorganism that lives inside the plant and is able to degrade contaminants that are taken up by the plant. And as a result, these plants can now grow on polluted sites. They will take up the contaminant, and then they will break down the contaminant to harmless water, CO2, and in certain cases nitrogen compounds. So it's a complete breakdown of the contaminants with no negative impact on the environment. It is also very important to know that when you create these organisms, this is not using genetic modification. So in Europe, for instance, there's a very strong opposition against GMOs. But microorganisms that we were creating this way are not considered as GMOs and can be applied. And actually, together with my colleague Jacob van Gonsveld at the University of Hasselt in Belgium, we are now applying this technique full scale in Belgium to actually do remediation of groundwater and soils contaminated with persistent organic contaminants.
So how are and you've also, at least that's what the website said, you've used microorganisms to brew hydrogen.
Yeah, so I've been working in several fields. So one field is environmental remediation. Another field is actually biofuel production. And that specific work that you're referring to was to look at thermophilic microorganisms. So these are organisms that can live at very high temperatures, and in this specific case also at high pressure, where we were using the microorganism to directly produce hydrogen from biomass, and then in the same situation use the hydrogen for further reactions. In addition, together with a group at the University of Ghent, also in Belgium, we did work where we are using microorganisms to produce in the soil the hydrogen, and then also have these microorganisms produce catalytically active nanoparticles, and these particles will then take the hydrogen and use it to break down organic contaminants in the soil. And again, this is also a process that is now being used in the field in Belgium to deal with certain very persistent contaminants like PCBs, which are also a big problem in the United States.
Mm-hmm. Mm-hmm. How do you plan to apply this to the new job here, to the new position, to the new center, and what are your plans on establishing this center? What do you need to have the center run the way you would like to have it run?
Yeah. So I started here at RTI the middle of January, and created together with Dr. Jenny Hunter-Severa this new Center for Agricultural and Environmental Biotechnology. And in this center there are 5 major areas of research. The first 2 areas we already briefly addressed. So it's environmental remediation, using plants and microorganisms to deal with persistent organic contaminants, but also to remediate large areas where you can't use classical physical chemical engineering techniques. Then in addition, biofuels and the conversion of biomass into biofuels, but also linked to this waste valorization. So for instance, anaerobic digestion of municipal waste or of animal waste. Also the growth of biofuel feedstocks on marginal land as part of land management. And when I was working at Brookhaven, one of the projects that we were having was actually to use plant-associated microorganisms to grow biofuel feedstocks on marginal lands. And then together with a colleague, Lee Newman, who is now at ESF in Syracuse, we also started looking at the role of these microorganisms in the sustainable production of agricultural crops. And this is really something that this new center is very interested in. So in addition to the 2 previous areas, the main focus of CAB will be on sustainable low input agriculture, where we are using microorganisms as ways to introduce systemic drought tolerance and systemic resistance against diseases in important feed crops, but also the production of produce that is of better quality, so basically better tasting produce. And I think we all have experienced that when you buy tomatoes that are grown in greenhouses, you say, they don't taste the same as the stuff I get from the garden or which I buy in the shop during the summer. We want to address that, so to get produce of better quality and better tasting. Then we are very much interested in food safety and food security, especially to look at where can potentially pathogenic bacteria enter the food chain. How do they live in association with plants? What is their risk of survival? How are they being propagated during the transport process, etc. And to see how we can minimize these risks. And then a third important area of interest is ethnobotany and medicinal plants. So in the United States we are spending about $800 billion on drugs that are produced by the pharmaceutical companies every year. But in a way this is heavily subsidized by the health insurance companies. What people don't realize, we are also spending about $400 billion every year on natural supplements. And so this is a very important market, but if you look, there are very few standards. So this is one of the fields we are very interested in, and to work with the industries in North Carolina, in the Appalachians, West Virginia, etc. And one of the projects we are starting to work on now is how can we domesticate some of the plants that have important medicinal properties that are currently being harvested from the forest. But of course, it has a risk that people are over-harvesting, not only in the United States, but also in other countries. So how can we protect our natural resources by bringing these plants into cultivation, but at the same time, how can we make sure that these plants still have the same quality as those that we harvest from the wild? So that's also an important field of interest. So these are the 5 major research areas. And then in support of all of this, we are very much pushing now to get in place a strong computational biology group that would be specialized in metagenomics, predictive metabolomics and microbial genomics, but also be a focus point for the integration of complex omics data with metadata. For instance, you can think of, well, for instance, one of the projects we have now is to look at the microorganisms that live in association with the Merlot grapes on Long Island. I lived for 11 years on Long Island, so I have very good connections there with the local wineries. And with some of these wineries, we are now looking at what are the microorganisms that live in association with these grapes. But in order to understand the differences, it's also very important to have what are called the environmental metadata. So we know exactly, thanks to the information these wineries are providing us, what is the soil type, what is the climate type, the water, how much fertilizer are these grapes receiving, when do they spray them against pesticides, etc. So the trick is not going to be to do sequence data analysis. Everybody can do that. The real issue is going to be to have these extremely large data sets, to integrate them and to draw meaningful conclusions, and to do it in an automated way, because these data sets are getting so big now that it is impossible for basically the human brain to deal with all of that.
Do you have, what are your plans on computers to use for the number crunching? Do you have the equipment here, or are you planning to use another computational facility, or how are you planning on doing that?
So it's a very good question, because the problem is for the moment that genomic data sets and metagenomic data sets are growing in an incredibly exponential way. When we were talking about the bioinformatics revolution a couple of years ago, it was that every 6 months the capacities of microprocessors will double. At this moment, the capacity of sequencing, which is in other words the technology that is being used to look at the genetic code, is increasing 100 to 1,000-fold every year. And to place it in perspective, it took the Department of Energy with NIH 10 years and $2.9 billion to sequence the first human genome. Mm-hmm. Nowadays, with the latest high-throughput sequencing machines, we can sequence in one reaction the equivalent of 10 to 15 human genomes for a fraction of the cost, maybe $30,000 for the entire process, which means less than $3,000 for covering once the human genome. So it's a completely different game. So sequencing has become a commodity. Mm-hmm. It's data analysis and data integration that is really going to be the critical success factor. So the data sets are growing exponentially, and how do you deal with that? Well, RTI wants to invest in this. We are seriously looking now with our ITS partner what kind of infrastructure we're going to need for that. But it's very well possible that we'll have to reach out to external sources, for instance, cloud computing, maybe the use of a supercomputer. We don't know yet. And I'm not a specialist in this field. It's really up to the specialist to decide at the end, this is the infrastructure we'll need. And then it's up to the management to make this happen. But we are very committed to make it happen. And we also think that this will be a great resource for the Research Triangle Park area to have in place. And we are very much looking forward actually to collaborate as well as with the industries, as well as with academic partners in this field.
What are you planning on doing if you're trying to experiment with feedstocks and produce and medicinal plants? Are you actually planning on growing them here? And if you are, how are you going to do that?
So for the moment, RTI doesn't have a greenhouse facility here on site. We have planned growth chambers, but again, this is an infrastructure investment we are making now, and we are in the process of planning of building here on site a research greenhouse facility that will be used for this work.
How big we're talking?
Originally between 3,000 and 4,000 square feet for the greenhouse itself, but with the possibility to actually expand it with future needs.
Okay.
And again, it's important to notice that most of the work, if not all of the work we would do in this greenhouse would be non-recombinant DNA work, so natural plants and natural microorganisms.
When do you hope to have sort of the rudimentary center set up, with the computing facility, where to go, whether you're going to go with the cloud, whether you're going to use somebody else's supercomputer, whether, you have several options there.
Yeah.
Have the greenhouse set up. When do you think you're to the point where you're really off and running and you actually can do work rather than build it up?
So actually we have had a very aggressive hiring plan, and a lot of the key staff will start before, at the latest, the 1st of October of this year. And for the infrastructure, so initially when you do work on plant microorganisms, the work is always starting in growth chambers. We have that in place, or we have ordered more growth chambers. They also will be in before the end of this fiscal year, so before the end of September. So basically we are going to be operational for most of the parts October 1st.
Mm-hmm.
And then next year will be a transition year where we are still planning of doing additional hires.
Mm-hmm.
We hope to have the computational group in place and completely up and running within the next year. But again, the first people we will hope to hire definitely this year. And then the computational infrastructure also is going to be set up within the next few months. So we are ready for business, I would say, now. We have the people, they have signed the contracts, and everybody is just extremely excited to get this started.
How many people you hope having by October?
So in October we will be around 15 people, and this does not include the bioinformatics. It will be kind of semi-autonomous group. And then we hope to double it by the end of 2012, and then we hope to grow it in function of contracts and industrial projects over the next few years.
And the contracts you're hoping, are you RTI? Most of RTI's revenue comes from government contracts and grants. Is that a similar plan, or are you hoping to work more with industry?
Yeah, so the work we are doing at CAB is partially basic research. We think we need our own research, and we want to create our own IP portfolio. But then in addition, we are very interested in working with industry, and RTI has a very good reputation of working with industry. But then in addition, we are also very much interested in working with the International Development Group to do much more international work. So I would say We want to go to a broad financial basis, different sources, and not just depending on one source like government funding, to create a more financially stable basis for this center.
Mhm.